So we'll do a quick overview of the course and talk about identifying an unknown organism as we review. So for the final exam, your study guide, as always, is your homeworks, right? Especially keying in on those terms, right, and being able to spell them. And then your quizzes are, of course, an excellent study guide. Although they don't cover all the material, right? They're a good sampling of the stuff that you should know. Exam covers all exercises after midterm, right? So most experiments helped you identify your unknown, but some were standalone exercises that were not associated with your unknown. You still need to know those. So there was a huge list of ones that helped us evaluate our unknown, right? Doing a hang drop mount or a wet mount if you needed to determine motility. We did the motility stab. You need to know all aspects of that test. The fluorothioalkylate medium that helped us determine if your organism could grow anaerobically or not. Your phenol red broth to let us know if it fermented or utilized the protein based on the different colors and if it produced gas. We ran the methyl red and BP tests, right? These were also fermentation tests. BP had to do with the production of what, though? What did we test for the production of? Acetonin, which then they can convert into what? A common byproduct of fermentation. Alcohol. 2,3-butanol to be exact. Right? That was the only test we did that actually tested for an alcohol. Methyl red test, remember it's a pH test, so again, acids. And the phenol red was the only one where we could see gas if it was produced. Citrate test was to see if they actually could take citrate in, right? If they had citrate permease. They either did or didn't, and they either grew or didn't grow. Catalase test, remember that enzyme can do what? Turn hydrogen peroxide into what? Water and gaseous oxygen, which is why it does what? It bubbles. Mannitol, salt agar, remember that has what in it that stops the growth of most organisms? Salt, but more specific, a high concentration of salt, of sodium chloride, more than normal, right? The mannitol, if it can grow, it can do what to it? Ferment it, right? And produce acid, and therefore the pH indicator, which is the same one in the phenol red broth, it's phenol red, turns what color when acids are produced? Yellow. It's the yellow plates in front of you. The Columbia CNA with 5% sheep's blood agar, what's the CNA stand for? 2 antibiotics, right? Colistin and nalidixic acid. I promise I won't make you spell them on the test. But you still need to know them, be able to identify them. And so those 2 antibiotics stop the growth of what? Or what does it allow to grow? Gram-positives will grow, it kills gram-negatives. And then if you had a gram-positive and it grew, and it changed the red blood cells, what if it was alpha, what would it have looked like? It was greenish, right? Because this is incomplete lysis of the red blood cells, where beta, complete destruction, right? And you can see right through those plates. What does gamma mean? No hemolysis. And then the eosin methylene blue plates we have just contain the sugar lactose, right? So we can tell lactose fermentation of what type of organisms, gram-positive or gram-negative? Gram-negative. What kills the gram-positives? The dyes, the eosin and the methylene blue, which also are going to create the color changes only the acid production from the fermentation of lactose, right? So all these we should hopefully be pretty familiar with. Going over your unknown report will really help you. And I'm going to grade them and give them back to you guys next week. But again, the exam is also next week. But hopefully you're on the right track. So standalones, standard plate count. And as I said, I'm going to try and do a quiz for you guys so you can get a review again of that. And we'll talk about that in this lecture too. Antibiotic sensitivity, remember the Kirby-Bauer method that we did? You need to be able to, and again, result table we would give it to you, just like we did on the quiz. By no means expect you to memorize how big the zone has to be for the different antibiotics, right? We would give you that table. You just have to know that you have to use it, right? And how to use that table. Remember anaerobic jar, we didn't actually do, but we discussed it, and you had quiz questions on it and homework questions, so make sure you review that and understand the principle of that jar and how it works. We're going to do today, one of my favorite experiments, we're going to do bacterial transformation using the P-glow system. So we're actually going to be using a designed plasmid circular piece of DNA to transform our bacteria. So our E. coli is going to go from plain old boring E. coli to ampicillin resistant, glowing under UV light, green fluorescent E. coli. So you guys unfortunately won't see that until tomorrow or next lab on Monday. I say tomorrow. How many of you guys are in my lab class? I mean my lecture class. Just Sydney. Seems like there was more of you guys. So you'll get a preview, because I'll bring it to class on Thursday night. So, and then the very last lab, we're going to do an ELISA, and we're going to do what's referred to as an indirect ELISA or an antibody capture, where we're detecting the presence of antibodies, actually using antibodies. So that one's pretty cool. That's one you'll read about for homework, and you guys will actually do next time. Another one of my favorites. So, you should also understand the process of identifying an unknown organism, which is what you just did right now, right, for some more practice other than doing it on your actual unknown. So, all the steps you perform to be able to identify your unknown will be on the exam. You need to evaluate stain slides, as I said, right? A negative stain, you got to be able to tell me the shape of the organism. You got to tell me any arrangement that you see, the terminology. You need to identify if you see a capsule or not. Gram stain, you need to know if it's positive or negative, acid-fast or non-acid-fast, and whether there are spores. So there'll still be plenty of microscopes for me to set up for your final exam. And so, as I said, shape and arrangement is important too. And you can pretty much tell that on all slides, although one of the main reasons why we have you guys do the negative one, it seems to come out best for trying to determine that shape and arrangement. So given a table, again, that table that you have in front of you will be given it again of known data for a set of organisms, test results of a pure culture of an unknown organism you need to be able to identify, right? So we have some practice problems. In addition, if you were given a table of data and it's not, we don't give you enough information, you still need to be able to narrow it down to what additional tests you would need to do to confirm, right? And so one of the ones, two of them problems actually you were given, right? You only could narrow it down to 2 organisms. Then you had to decide what test, and when we say test, we're talking about from the table, right? And as far as plates go, we're not going to ask you, like I said, Bacillus cereus and Bacillus subtilis are beta hemolytic. Y'all, it's taken me years to memorize that. We're not expect you guys to memorize that for those organisms. So the first example, this one's pretty easy, right? One of these results should really jump out at you, and that was the acid-fast positive. Should lead you right to Mycobacteria smegmatis. But if you look at all the rest of the information we give you, you would still narrow it down to Mycobacteria smegmatis. Mycobacterium. Excuse me, bacterium. Got to work on my pronunciation. So for the next one, right, we give you these following results, and you're only narrowing it down to 2 organisms, right? Which 2 did you guys get? The 2 staphs, right? Staph aureus and Staph epidermidis. Epidermidis. Right, so, right, gram positive, we can eliminate the whole top half of the chart, right? Because all those are negatives. And then phenol red, right, it was A-, which means acid, no gas, right? So we can eliminate it down to just these 6 organisms. Then we take into account the citrate result, which was negative. We can eliminate all the bacillus. When we take into account the catalase, which was positive, we can eliminate Enterococcus faecalis. I say it right? Faecalis. Right? And we're left with the 2 Staph aureus. So what result for these guys is different? What tests had we not performed yet? BP, right? The BP is what we could narrow it down to, but we would want it to be just one test, right, to determine the difference. So we would also use one of our selective media. Which one's going to help you out with this? MSA? Is that it? Yeah. Yeah. Yep. You could do that one. Right? And Staph aureus does ferment where Staph epi doesn't. There's actually one more that you could do, but we wouldn't ask you guys it. The blood agar. Staph aureus is beta hemolytic. But knowing that information about mannitol salt, you do need to know. So technically that one had 2 correct answers. Okay, so the next one, again, this one actually could narrow it down to a single organism. What did you guys get? Yeah, Serratia mercescens. Right, mercescens? So, again, it was gram-negative, so we can eliminate all the positives right off the bat. That cuts the field in half, just about. Methyl red, it's positive, right? So that brings us down to 4 organisms. And then citrate knocks it down to either Klebsiella pneumoniae, right, or Serratia mercescens. The phenol red helped us narrow it down to the single one because this one is not a gas producer. Now for the next one, you ended up with 2 possibilities again, right? Which 2? 2 bacillus, right? Bacillus megaterium or Bacillus subtilis. So the spore is a dead giveaway when it comes to bacillus, right? Because those are our spore formers. So we can eliminate it down, bam, with that one result down to 3. Right? Take into account our methyl red and anaerobic growth. We narrow it down to just megaterium and subtilis. These guys, again, only one test result is different for these 2 organisms, right? And that is the VP. So what one test? The Voges-Proskauer test. What media would help us out? Blood agar, right? And again, we don't expect you guys to memorize this one, but the Bacillus cereus would be beta hemolytic, and the megaterium would be gamma, which is no hemolysis.
On the test, on the final, are we going to have to spell out that?
Oh, Voges-Proskauer? No, you could write VP. Okay, so the first half of this course, right, we did ubiquity of microorganisms and light microscope and examination of eukaryotes. You learned common aseptic transfers and did streak plate method. All that stuff is basic stuff. The stuff that you're probably most excited about that you won't see on the final is the eukaryotes, right? And then we stained, which we did again, right, on our unknowns, right? So you learned and then you practiced. So that's what really carries over, right, is the staining. So then we had a few additional experiments other than what we did on our unknown, as we said, standard plate, antibiotic sensitivity, P-glow and ELISA that we still have left to do, and anaerobic jar. So this is a good chart, right? charts are really good for when you're studying, right? Trying to organize information. And so remember that cells are negatively charged. Their phosphate groups accompanying their DNA and their lipid membranes make them negatively charged. So remember, these dyes, these basic dyes, are what charge? Positive, right? And they're attracted to our cells. So they stain the cells themselves. And a very simple stain, just using one stain, typically a basic stain. Where negatively charged dyes or acidic dyes repel, so they stain the background. And the two that we used were nigrazine and Congo red. And then our other, so those very simple stains, at the most you're going to be able to tell cell morphology and arrangement. The rest of the stains we did gave us a little bit more information about our organisms, right? So, which staining procedures are differential that can tell us more than just cell morphology and arrangement? You can still tell arrangement, right, and morphology with these stains, but it tells you more information. All the rest of these, right, would be considered differential. They're going to tell us something structurally different about these cells. So, which are structural, actually allow us to identify specific structures. Capsule and endospore, right? Those are distinct structures. The capsule's outside the cell, right? The endospore is within. And remember, we used a particular method. We used the Schaefer-Fulton method. So that's why we have green spores and we have pink to red colored cells. You can get a positive or negative result for both of these, right? Although a spore isn't always a true negative, right? Because maybe it didn't produce spores yet. The other two, right, are going to tell us about what's the makeup of these cells. So remember, gram-positives versus gram-negatives. It's going to tell us that thickness of the peptidoglycan layer. Remember, the gram-positives very thick layer, gram-negatives very thin, and they usually have those two membranes. Acid-fast is a very specific composition of the cell wall, right? That cell wall contains mycolic acid. And we know one genus of bacteria that have this, and that's your mycobacteria. That's why when we get a positive acid-fast, right away you should think mycobacteria. Right? That one's really easy. Most of you guys had a negative result, right? Because only one person had mycobacteria. Alright, so for standard plate count, why do we do this? To determine the concentration of viable cells, cells that will actually grow in our original cell culture, in our original concentration. We measure this in CFUs, which stands for colony-forming units, right? We're not actually counting cells on that plate, we're counting colonies. And that could have aroused for more than one cell, right? So we really can't say cell. What did we do? We did serial dilutions, right? So we need to understand, right, how to do a dilution, how much we dilute it, and then we only count plates within 30 to 300. That's the statistically accurate range. Less than that, less than 30 is really too few. And more than that, I don't know about any of you guys, but I would want to count more than 300. And it would be really difficult to count that many cells. So, important things we need to know is the dilution factor, right? What level of dilution did we do for each dilution step in the series, and then what the final dilution, that total dilution of that tube is. And then we need the volume that we transferred out. Right. So remember, each individual tube has a dilution, and then we figure out the dilution of the tubes. So we have our colony forming units divided by the volume that we plated, multiplied by the dilution factor of the tube that we took the sample from. And you guys are somewhat going to be doing that today. Because you're going to be taking set amounts out of your tubes today and plating them. So theoretically, if you guys get them spread out real good, you could come back and do a colony count and figure out how good our transformation was today. So 3 things you need. The dilution factor of the tube, right? That total dilution factor of the tube that you plated, the volume you plated, and the number of colonies on that plate, which of course have to be in range. All right, so here's an example of where they moved from the original culture, 0.1 milliliters into 9.9, which is a 100-fold dilution. Then they continue with another 100-fold dilution. Then they did a 10-fold, 10-fold, 10-fold dilution. But remember, if you do 100 here and you move from here to here, this is a combination of 100 by another 100, right? So that's 10 to the 4th dilution in this tube. That's the total dilution of this tube. And when you do exponents, it's easy because you can just add them. But remember, when you multiply is how you would get your values. But when you multiply exponents, you just add the exponents, right? So once we know that, and if you know the volume that you plated onto the plate, so in this case, 1/10 of a mil. We're doing it all in milliliters here. Then we have all the information we need except for colony count. So you count up your colonies. These are the numbers that you get. Which one's actually accountable? 128, right? It's between the 30 to 300, plate Y. What is the titer of the original culture? What's the concentration? So what are the 3 numbers we need? We need the colony count, right? So we need 128. And then we're going to divide that by the volume, which is 0.1, and multiply by the dilution factor of that tube, which is 10 to the 6th. So in this case, written in scientific notation, 1.28 times 10 to the 9th. So 1 billion CFUs per milliliter of this sample. If the original culture was 20 milliliters, we just multiply by 20. That will cancel out the milliliters and give you just the total CFUs. Right? So in this case, it's going to give you 2.5 times 10 to the 10th CFUs. That's a lot of bacteria. And that could even be on the low side, right? If the colony arose from more than one cell. So antibiotic susceptibility, why do we do this? To determine if an organism is sensitive, right? Or susceptible to a particular antimicrobial. Remember, antimicrobials can be natural or synthetic. If it's natural, it's actually called an antibiotic. That's the true definition of a naturally occurring antimicrobial substance. We looked at ones that were bactericidal, will actually kill the bacteria. Others that were bacteriostatic, which is inhibit growth and give your immune system a leg up on fighting that infection. We used a particular method, right? We started with a particular density of cells. We worked with a particular type of agar. It's actually Mueller-Hinton 2. Everything is standardized in this test, right? We started with an approximate cell number, spread it out over the plate, we added the antibiotics, and then the antibiotic discs diffused into the media and affected the bacteria, right? Then we measured those zones in millimeters and compared them to our chart. Remember, different organisms respond differently depending on their chemical makeup. Like gram-positives are more susceptible to penicillin than gram-negatives because penicillin affects the cell wall. So it diffused out, right? We had those clear zones that we measured. You must use the interpretive chart. We will provide you with the chart, right? But usually a large zone will mean it's sensitive, but you still got to measure it, right? Intermediate, somewhat sensitive. If there's a small or no zone, it's probably resistant. But again, you would measure these to determine. Remember, we had controls in a lot of the tests that we run, including today, right? So you have the possibility of false negative results. This is usually a sensitivity issue with tests when you get false negatives, right? The test isn't sensitive enough. It's one of those things that we'll talk about again in the ELISA test that we'll do next time. False positives, it's usually a specificity issue, right? There's something else there that's causing it to become positive. For some of you guys' tests, it may have been a contamination issue, right? There was something else there that was causing it to be positive, right? So we want to be aware of these limitations of our tests, sensitivity and specificity issues that can lead to these false, negative, and positive results. So what Peter did here was just go through, right, each one of the tests. Why did we do it? What was the method? What are the results? Which is what you guys should do as you're studying, right? One of the things that will help you is go through each one of these. We have the scientific method in each one of the PowerPoint presentations, right? Know what your results tell you about each one. So I'm not going to go over each one of those. Important thing with fermentation, right? We're detecting acids, alcohols, and gases. Some of the tests we did can detect only one of these products. The only one that could detect two was the phenol red, because we could detect gas and acid, right? And in fact, that was the only one where you could detect acid. Lots of times we were looking for acids, and you guys remember what test was the only one we did for the detection of alcohol from fermentation? The one you don't have to spell, you can use the abbreviation. BP, Folks Press Coward. That was the only one. Right? So, let's see. So make sure you go over each one of these tests. And I may do that next time. He's got a lot more in here that I'm not going to go over. Yeah. Okay. So this is a prelude into what we're going to be doing today. So bacterial transformation is what we're going to do today. So we will make glowing E. coli. And then I love this acronym that he has here, and I want you guys to, why I went to this slide is to think about this as you're reading through the ELISA for next time. We make what he likes to call an antibody sandwich. Right? So we have layers stacked up on each other. So we're going to start with a microtiter well, little tiny wells, and we're going to attach an antigen to it, and then we're going to attach an antibody to it, the primary antibody, and then we're going to attach a secondary antibody, and what's special about this one, it has the enzyme linked to it. So if all these things are present, right, if we get this sandwich of antigen, antibody, antibody, when we add our colorless substrate, we'll get a color production because the enzyme's going to convert it, and we'll get this blue color. Washes are performed in each step so that anything that does not bind gets washed away, and it doesn't interfere with the accuracy of our results. So a direct ELISA is where you're testing for the presence of an antigen. So usually you're getting a sample from your patients that contain an antigen. And then we're going to use a purified antibody to detect the presence of that antigen. And then the secondary antibody helps us detect the first antibody. And again, is special because it has the enzyme linked to it. And these can be bought, right? There are kits that can be done. So that's the question. The question we're going to be asking, and to give you an idea of some type of tests out there, there's lots of bacterial tests. Kate has done this recently with her daughter. I bet they did the rapid strep, didn't they? Right? So that's an antigen-based test where they're using antibodies to detect the presence very quickly within minutes if that antigen is present. They can do this for viruses, for HIV, they can do it for flu, for the rapid flu test. They can use hormones as an antigen, so the pregnancy test you buy over the counter, those are this technology. We're making an antibody sandwich. Drug tests, some of them use this type of technology as well. What we're going to do is an indirect ELISA. And the only difference is that in the sample, it's going to contain the antibody. So we're going to start with purified antigen, and then we're asking, does the person have the antibody specific for the antigen we're starting with? And then we'll use our secondary antibody to detect the first antibody and create our color change. So same thing, the only difference is again, what's the question? In this case, do they have the antibody? So again, lots of viruses, we can detect whether someone has had that viral infection based on whether they have the antibodies in their sample. When you take your dog to the vet and they take a blood sample and they're testing it for heartworm, they're doing an indirect ELISA. They're seeing if your dog has made antibodies against that worm. For hepatitis, they're seeing if you have antibodies against that. And the problem with that is you can also produce antibodies when you've been vaccinated. So that doesn't necessarily tell us whether you have that infection or whether you've been vaccinated, because you get the same result. You get antibody production. Where if we tested for the actual antigen, the actual virus, we would know if you have the infection currently. So that's the end of our review. Oops. No. Yes. No. No.